Dear ladies and gentlemen, thank you so much for joining me today for my little speech about the microRNAs and their incredible potential and power as the future of the precise diagnosis and more effective therapeutics. BioVendor is a family of biotechnological companies with a common aim to introduce novel and innovative scientific methods to everyday diagnostic life. We focus on speeding up the process of a technology transfer to the diagnostic practice, and the main reason why we do so is to deliver the world better, more specific biomarkers which can help with the early detection of the disease, proper treatment design, and overall personalized healthcare. The key discipline to the personalized medicine is the clinical genetics, and we have several tools which could be selected. The DNA is a perfect nucleic acid for the detection of hereditary diseases. It provides us with a direct answer and help us to initiate necessary treatments, lifestyle adjustments, and precautions. The DNA encodes for a lot of information, like various predispositions or risk factors. This can be very useful as, for example, knowing the fact that you are genetically prone to lactose intolerance, you can limit the lactose intake and minimize the risk of intolerance manifestation. On the other hand, the DNA does not really reflect the current situation in your body. That means that knowing that you have the BRCA mutation doesn't necessarily mean that you have the cancer at the moment. This is the reason why geneticians all around the world were searching for a better, more accurate template which would really reflect the actual state of your body. And so they started using the messenger RNA, which indeed provides you with the answer regarding the actual state of your body. The messenger RNA is the expression product of the DNA, and it really provides you with the potential of detection the tumor cells very early. On the other hand, messenger RNA is extremely unstable. It's very tricky to work with, and you need to go for a tissue sampling to make sure to identify its presence. Therefore, there is a new space for novel biomarkers like microRNAs. The microRNAs are also the product of expression of DNA, and the microRNAs also can reflect the actual state of the body. They are very early biomarkers as you can really detect the differences of their expression from the beginning of the disease. And they are really great because they're very stable and they are present in the biofluid, so you don't need to examine the endangered tissue. You can just search for its expression in the blood or tears, sweat, urine, or whatever biofluid you go for. As I mentioned, the microRNAs are expression products of the DNA. In the past, we believed in the central molecular dogma, which claimed that the DNA is transcribed into the messenger RNA, which is translated into the protein, and that's it. So we expected the whole DNA to be protein encoding. But once the Hugo project was finished, we find out the very opposite, that only very small part of the DNA is actually protein coding. And the rest of it was called junk DNA for decades, because we expected not really big meaning of these sequences, but we were so wrong. These other parts, so-called junk DNA, were coding for a lot of regulatory products, and one group of them is non-coding RNA. The non-coding RNAs are divided into families based on their size. So there is a group of long non-coding RNAs which are responsible for heterochromatin and euchromatin formation. And they actually regulate also the expression of the other group of non-coding RNAs, which is short and mid-sized non-coding RNAs. This family includes microRNAs, PV interacting RNAs, small nuclear RNAs, and other short non-coding RNAs, which are responsible for post-transcriptional and post-translational regulation of a genetic expression. So these small molecules are the key orchestrators at the molecular level. So there are a few more characteristics about the microRNAs I would like to talk about. The microRNAs are very stable, and the great thing about them is that they are usually produced by the original cells, but then they need to travel to identify the effector cell. And for that, they can either use the exosomes, microvesicles. They can be a part of the protein or a lipid complex, but they can also travel on their own. So from the basics of their molecular effect, they need to be stable in the biofluids. Another great thing about microRNA is that they are tissue specific. That means that every tissue in your body has a certain subset of microRNAs which are expressed, and this expression profile can be altered by any pathology happening in that particular tissue. If we look at the in situ hybridization of the murine embryos, you can see that while the microRNA1 is highly expressed in the heart and the cardiovascular system of the embryo, the microRNA 124A is strongly expressed in the brain of the embryo, in the neuronal tube of the embryo, and this is a big difference. So the great thing about microRNA is that if you know which microRNAs to target, you can identify specifically which tissue is suffering with what specific type of pathology. And the last characteristic of the microRNA I want to talk about is that the microRNAs are the key orchestrators of every molecular mechanism happening inside the cell. They monitor the DNA replication, chromosome segregation, cellular division. So if you think about any signaling pathways inside the cell, the microRNAs are involved. So this is, for example, the signaling pathway of P53, which is the world's most studied and best known tumor suppressor. And you can see that the microRNAs act at all the levels of these signaling pathways. So they regulate the immediate response to any change at the molecular and cellular level. Therefore, they are so early biomarkers for any pathology you can think of, because they act as the molecular regulators of this pathology and they respond to it. So talking about the microRNAs and the pathologies, we know that they have incredible potential for the early diagnosis of various cancers, cardiovascular diseases, neurodegenerative diseases, metabolic diseases, or even inflammations. So therefore, they become in the spotlight of the key opinion leaders worldwide because we know of all of this potential of their diagnostic strength. Because of all of this great potential, BioVendor established microRNAs as one of their key projects back in 2017. Ever since then, we purchased and upgraded 2 quantification platforms, and we have developed over 150 individual assays. BioVendor encounters a group of specialists working at the Research and Development Department, product manager, and a team of hardworking people who are very enthusiastic about the microRNA. At this moment, BioVendor offers a complete solution for all different microRNA projects, started with the sample isolation kits, as working with any nucleic acids is always challenged by its proper isolation. We have manual kits for different sample types, and we even have an automatic solution. Also, sometimes you are not sure which microRNAs are really the ones which your pathology or project or phenotype should be focused on. So we have a group of screening panels which you can use to identify the proper targets. Once you know which microRNAs you want to quantify, we can develop customized assays to your particular targets for the proper quantification. And BioVendor also distributes various microRNA-based diagnostic systems, which some of them already have the CE-IVD certification. MicroRNAs were discovered back in 1993 by a scientific group of Victor Ambros, and ever since we know them, it was a big challenge to properly quantify only the microRNA of your interest. One of the problems is that microRNAs are very short molecules, so they are only about 19 to 24 nucleotides long. Mature microRNA do not contain any common sequences like the poly-A tail or the cap. They're quite rich in a GC content, and the mature sequences can be nearly identical in the family members. So you can have a lot of microRNAs with very similar sequences. And you need to make sure that your quantification assay really properly distinguish between them. Another problem is that the microRNA has its own synthesis process. So the mature microRNA is a part of its precursor and the primary variant of microRNA. And you also need to make sure that your assay is quantificating only the mature variant. A lot of producers were trying to solve the problem with microRNA quantification by unspecific reverse transcription. So they use the reverse transcription primer, which is, for example, a stem loop system, but that means that there are only 2-3 nucleotides which are really specifically targeting the microRNA of your interest. So you produce very unspecific cDNA library, which then enters the qPCR cycle. Another version of the approach was artificial polyadenylating of the microRNA. But that means that the poly-A tail is added to any microRNA, any mature microRNA in your sample. And then again, you create very unspecific cDNA library, which then enters the real-time quantification with the primers which are partially specific to your microRNA and partially specific to the poly-A tail. And this is where the BioVendor makes a difference. So our first approach is called two-tail PCR assay. And the biggest difference is that we have used a specific hemiprobic RT primer, which covers as well the 3' end of the microRNA as well as the 5' end of the microRNA, which means that we cover almost 80% of the final targeted microRNA at the cDNA reverse transcription level. So we create very specific cDNA library, which then enters the qPCR cycle with specific primers targeting the rest of the molecule and the 3' part as well as the 5' part. The superior specificity and sensitivity of the two-tail PCR assay is achieved by introduction of the 5' part of the target microRNA recognition. So as I mentioned, our reverse transcription primers are recognizing both 3' end and the 5' region of the microRNA of the interest. The same is true for the qPCR. So if we look at the reverse transcription, there has to be 100% match between both hemiprobes, both arms of the reverse transcription primer. If there is a mismatch or not a complementary region of the attached microRNA, there is no reverse transcription. Then, during the qPCR. Our qPCR primers covers over the region which was not recognized during the reverse transcription. And if there is any mismatch during this qPCR cycle, the polymerase refuses to amplify this region. That means that because of usage of both 5' ends during the reverse transcription as well during the qPCR, our two-tailed PCR has this superior sensitivity and specificity. So we are very sure that we only quantify the microRNA of our interest and not its family members, isoforms, or other isomers. To compare the performance of mentioned approaches, we quantified different family members of the LAT7 microRNA family. And you can see that the approaches which were using a general polyadenylation of the targets have very big problems with recognition of similar family members, which only differs in 1 or 2 nucleotides. Our two-tailed qPCR was capable to distinguish between the different family members, mostly because we are very specific from the first reaction, which is the reverse transcription. Besides its excellent specificity and sensitivity, the two-tailed PCR is also very easy to be used. So you only need a general qPCR equipment to run the two-tailed PCR. So you literally need a thermocycler and the real-time system. And the two-tailed PCR uses a standard cyber-green FAM channel for the detection. So majority of the RT systems can work and are compatible with our two-tailed PCR. To summarize the two-tailed qPCR assay, I would like to highlight that it is extremely sensitive and specific. It is very robust and it's great and specific because it doesn't add any artificial sequences to the target microRNA. We know that it's only quantifying the major variant of microRNAs. And not only microRNAs, we have also experienced the possibility of quantification of other small non-coding RNA sequences quantification. So we succeeded with development of assays for microRNA, PV interacting, or small nuclear RNA quantification. It's very easy to prepare, to run, or to work with, and at this moment we can develop multiplex reverse transcription reaction for several of your targets. You only need a basic library equipment, and the great thing is that whatever target you have, we can develop a customized assay for it. Therefore, even if you can't identify your targets at our web pages, we can develop the assays customized only for your project. Now, I would like to introduce very briefly a second platform which BioVendor offers for the microRNA quantification, which is called miREIA, and it stands for microRNA enzyme immunoassay. miREIA is very similar to the ELISA and differs mainly in the first step. So the miREIA starts with the hybridization of a single-stranded DNA oligo, which is labeled with a biotin. This single-stranded oligo is complementary to the microRNA of your interest. The microRNA is also single-stranded, so during the hybridization, it creates a heteroduplex, which consists of one single-stranded DNA oligo and one microRNA single-stranded molecule. So you have a DNA-RNA heteroduplex. This particular heteroduplex is identified and recognized by the antibody which is captured to the microtubule plate. And this antibody only recognizes a perfect heteroduplex. So if there is a nucleotide mismatch, it creates a secondary structure, a bubble. And this bubble doesn't allow the antibody to recognize the heteroduplex. So the antibody recognizes only perfect heteroduplex, which means that it only recognizes the labeled microRNA of your interest. The rest of the procedure is, as I mentioned, ELISA-based. So you just add the substrate and you do the colorimetric quantification. miREIA can be very well applied in any immunology laboratory, because you really need only a basic instruments which are necessary for a standard ELISA quantification. So you need the absorbance reader, some kind of a basic thermocycler, centrifuge, and the temperature incubator. miREIA is an excellent quantification platform, which unfortunately also have its cons. So miREIA has a detection limits which are slightly less sensitive in comparison to the total PCR. So you need to keep in mind that miREIA is a perfect assay if you're working with a highly expressed microRNA. Another great thing about miREIA is that it is, as I mentioned, ELISA-based platform. So the application and generally the usage of this assay is very easy. It uses a specific RNA-DNA hybrid detective antibody. So it really only detects those hybrids which are perfect. It is validated on the synthetic microRNA, and it's capable to do the absolute quantification of your target. It doesn't include the reverse transcription or the amplification, which means that it has this detection limit, but on the other hand, it doesn't introduce the mistakes which enzymes like to introduce. It has very comparable performance to the other reverse transcription qPCR techniques. At this moment, you can multiplex up to 2 targets per one plate. And it is quite sensitive method. So you can really distinguish between different family members. Again, our R&D team is capable to develop a customized assays based on your requirements, based on the requirements of your project. So don't hesitate to contact us, and we can prepare the assays particularly specific for your targets. Sometimes the biggest challenge is to identify the key microRNAs. The modern technology allows us to choose between many approaches. RNA-seq or NGS are the high through output techniques with a huge and large volume of results and raw data. It provides you with so much and so complete information that it might be too complete and it becomes confusing. To differentiate between the random changes introduced by the circadian rhythm or the basal metabolism or just by the cell-cell adhesion, and those changes which are particularly specific to your project might be very challenging. Our screening panels only focus on pathology-related microRNAs. The large panels, the cancer and the biofluid, are strictly looking at the expression of pathologic microRNAs. The cancer panel profiles over 350 pathological microRNA with over 30 controls. Those microRNAs are mostly strongly associated with the various cancer types and the various regulatory pathways of the key onco and the tumor suppressor genes. Biofluid panel is profiling over 170 microRNAs with more than 20 controls. It was established specifically to work and to be functional in the various biofluids like a human serum, plasma, or even the urine for the various pathological conditions. Osteomer is a quantitative panel which quantifies the microRNAs related to the bone metabolism. So the microRNAs from these panels are responsible for the bone regeneration and formation, for the calcium absorbance, for the osteoporosis risk and the fragility risk fractures. Thrombomer is the last microRNA profiling panel I would like to talk about, and this one is focused on the activity of a platelet in the human blood. So these novel biomarkers are linked with the platelet activity, which is adjusted with the various treatments, with the anticoagulation treatment. So if you're working on any project which can be specified by any of the descriptions I provided with these panels, we can use any of these panels to identify the microRNA which will be specifically influenced by your project, by your phenotype, your mutation, or your pathology. What we generally do is that we offer a special project which consists of two different phases. The first phase is including the knowledge panel. So that means that either we or you can run the knowledge panel, the screening panel, and identify those microRNAs which are really essential for your project. This first phase can be also offered as a BioVendor analytical and testing service. So you can supply us with your samples and we run the analysis and provide you with the full report and the raw data. Otherwise, it's also possible for you to purchase the knowledge panel and run them on your own. But you will never lose our guidance, you will never lose our supervision. You can always come back to us with the results and we help you to troubleshoot, to understand and to interpret correctly the results of your work. Once you identify the products of your interest, once you identify the targets which are significantly influenced by your project, by your pathology, we can produce customized assays either in the two-tailed PCR platform or by using miREIA platforms. That means that we can produce specific product based on your needs. It can also be multiplexed either at the reverse transcription step or by several targets within one plate for miREIA. And we guarantee the performance of both of these platforms as we always wet lab test every single assay. So every assay which is produced by our research and development department is always tested on the synthetic molecules and even within the biologic samples. So now, if you're considering the starting of microRNA-based project, you know that you can rely on BioVendor. We can help you with the proper selection of the sample type. We can help you with the accurate isolation protocol and the isolation kit for your project. We can help you to search for a special microRNA of your interest. We can help you to identify those microRNAs which are significantly influenced by your phenotype, by your project. And once you know which microRNAs you want to focus on, we can produce a customized quantification assays based on your preference or based on your project needs. Dear ladies and gentlemen, my name is Veronika Grešáková and I would really like to thank you for your attention. I hope the speech was not too complicated or hard to follow. And now it's the right time to start the discussion and I'm ready to answer your questions.